Hello friends, welcome back to my YouTube channel Journey with Vijay Kumar Shrivastav. Today I am going to present the topic Deterioration of Varietal Genetic Purity. Before presentation I would like to introduce myself. I am Vijay Kumar Shrivastav. I have done M.Sc. Agriculture with a specialization in Agronomy and presently associated with seed industry related to hybrid seed production. This video has been prepared considering the students who are preparing for various agriculture competitive examinations and other exams. Hope this will be useful to all of them. So let's move on. First, to understand what is variety and genetic purity. Variety represents a defined group of plant within a species having common set of characteristics. In other way, variety is a group of plant having clear distinct characteristics which when reproduced either sexually or asexually retain its original true characteristics. The main aim of seed production is to produce genetically pure and good quality seed. Now genetic purity. Genetic purity is the true to type nature of seed which is matching with its mother in all aspects related to seedling, plant and trees produced. This quality character is important for achieving the desired goal of raising the crop either for yield or for resistance or for desired quality factors. Deterioration of genetic purity. Why and how the genetic purity of a variety is lost or deteriorated during seed multiplication? The genetic purity of a variety or trueness to its type deteriorates due to several factors during the production cycles. There are several factors which are responsible for the loss of genetic purity during seed production listed by Kadam in 1942 are described as developmental variations, mechanical mixtures, mutations, natural crossing, genetic drift, minor genetic variations, selective influence of pest and diseases, techniques of the breeder, breakdown of male sterility and improper or defective seed certification system. So we will study in details about each responsible factors. First factor is developmental variations. When seed crops are grown under environments with differing soil fertility, under saline or alkaline conditions, different climate, different photoperiodic conditions, or at different elevations or different stress conditions for several consecutive generations, the developmental variations may set in as differential growth responses. It is therefore preferred to grow the varieties of crops in the areas of their natural adaptation to minimize developmental shifts. To eliminate or minimize such developmental variations, the variety should always be grown in suitable adaptable area or in the area for which it has been released. If due to some reasons like lack of isolation or to avoid soil-borne diseases, it is grown in not adaptable areas, then it should be restricted to 1 or 2 seasons and the basic seeds like nucleus and breeder seeds should be multiplied in adaptable areas only. Second factor is mechanical mixtures. This is one of the major source of contamination of the variety during seed production. Mechanical mixtures may take place right from sowing to harvesting in field and during processing operations in veg. And these factors which are cause of mechanical mixtures are like first one, contamination through volunteer plants of the same crop in the seed field. So these volunteer plants are the plant of same crop which are existing in field from previously grown season. Means these volunteer plants are germinated plants of the same crop which were previously cultivated. So we have to remove all volunteer plants to avoid any physical mixtures before harvesting. Second source is seed drill. If more than one variety is sown with the same seed drill, then it should be thoroughly cleaned, because there are chances of leftover seed in the bin, seed drill bin. So to avoid any mechanical mixtures, it should be totally cleaned. Third is growing through different varieties grown in adjacent fields. If side by side there are different varieties grown, so there are chances of contamination by mechanical mix. So we have to take all precautions that it should not be mixed. Fourth factor is the seed mixing during harvesting and threshing operations on threshing floor. We have to take all precautions during harvesting and during threshing operations. Threshing floor should be totally cleaned, and while harvesting we have to take care that from the side by side fields or whatever combines or threshers we are using, it should be not having the earlier leftover seeds. So the combine and thresher should be also checked and cleaned thoroughly, and there should be proper supervision before use. Other sources are also there for contamination, like gunny bags, seed bins or elevators, because these are the often source of contamination. So if the gunny bags are old, previously used, it should be totally cleaned. Seed bins to be clearly checked. There should be no any presence of seed of earlier lots or earlier other varieties, and elevators should be cleaned. Another source of contamination are during seed processing operations. All machines, these mechanical parts should be totally cleaned, and it should be supervised to avoid any mechanical mixture chances. To avoid this sort of mechanical contamination, it would be necessary to rogging the seed fields also critically at different stages of crop growth, and to take utmost care during seed production. Harvesting, threshing, processing, etc. are necessary to avoid such mechanical contaminations. In seed production, to maintain the genetic purity, rogging is very important. Rogging refers to the act of identifying and removing plants with undesirable characteristics from agricultural or seed production fields on visual inspection basis. These rogue plants are removed from the fields to preserve the quality of the crop being grown. And rogging is important practice in seed production to maintain genetic purity of seed. These rogue plants, undesired plants, are causing deterioration of genetic purity by two means. First, by contamination through natural crossing, and second, by mechanical mixture if not removed before harvesting. So rogging is very important and having the critical role in improving the genetic purity. Rogging at all stages of the crop in the field is an essential requirement to maintain the varietal purity, as it was at the time of release or notification of the variety. So there should be true to type quality maintenance. As rogging stages are concerned, rogging should be done timely and prior to any contamination. Rogging is done from vegetative stage to harvesting based on many parameters as morphological and reproductive characteristics. For most of the crops, the critical stages are vegetative stage, pre-flowering stage, flowering stage and maturity stage. Third factor of deterioration is mutations. These mutations do not seriously deteriorate varieties, and it is not of much importance as the occurrence of spontaneous mutations is very low. It is often difficult to identify or detect minor mutations occurring naturally. If any visible mutations are observed, they should be removed by roguing from seed plots to purify the seeds. In case of vegetatively propagated crops, periodic increase of true to type stock would eliminate the mutations. So these mutations are not one of the serious factors for causing the deterioration of genetic purity. Fourth factor for deterioration of varietal genetic purity is natural crossing. Natural crossing refers to the movement of pollen from one plant to the stigma of second plant where the fertilization takes place. Natural crossing can be an important source of contamination and varietal deterioration in sexually propagated crops. The extent of contamination depends upon the magnitude of natural cross-fertilization. The deterioration sets is due to natural crossing with undesirable types, diseased plants or off types. In self-fertilized crops, natural crossing is not a serious source of contamination unless variety is male sterile and is grown in close proximity with other varieties. On the other hand, natural crossing is main source of contamination in cross-fertilized or often cross-fertilized crops. The isolation of seed crops is the most important factor in avoiding contamination of the cross-fertilized crops. To overcome the problem of natural crossing, isolation distance has to be maintained. Increase in isolation distance decreases the extent of contamination. The extent of genetic contamination in seed fields is due to natural crossing depend on breeding system of a species, isolation distance, varietal mass and pollinating agent. The direction of prevailing wind, the number of insects present like honeybee activity and mass of varieties are also important considerations in extent of contamination by natural crossing. In seed production, to maintain the genetic purity of a variety, isolation distance is one of the important critical factors. What is isolation distance? Isolation distance is the minimum separation required between two or more varieties of the same species of the crop for the purpose of keeping seed pure. So, this isolation distance refers the minimum criteria of the distance or time required between two crops of same species to avoid any contamination between them. There are three types of isolation. First one is spatial isolation. Second is temporal isolation or time isolation. And third is physical or barrier isolation. What is spatial isolation? A spatial isolation or a spatial separation required between a seed field and other sources of genetic and mechanical contamination, especially between varieties of cross-pollination. Means a spatial isolation refers to the minimum distance which may vary crop to crop, variety to variety. So this minimum criteria has to be followed to avoid any contamination, and this is very important for cross-pollinated crops. Second is temporal isolation. Crop of seed production should be sown early or late by a margin of 20 to 30 days, which may vary depending upon the parental nature. Then neighboring fields of the same or other variety to avoid same time flowering and to prevent entry of foreign pollens in the field of seed production. Means this temporal isolation or time isolation is the required gap between two crops that it should not match same time flowering. Third one is physical or barrier isolation. It can be practiced by covering the floral part or whole plant. We can avoid the physical contaminants into seed fields, where we can use paper bags or cloth bags on the floral part, or we can use these tarpaulins to hindrance the pollen movement from one field to other field. There are factors which are affecting isolation distance, like pollination behavior of the crop, pollinating agents, pollen trails, wide pollinator or designated diseases, or type of seed production or class of the seed. So we have to maintain the proper isolation for the preservation of the genetic purity. Next factor of deterioration is genetic drift. When seed is multiplied in large areas only, a small quantities of seed taken and preserved for the next year's sowing. Because of such subsampling, all the genotypes will not be represented in the next generation and leads to change in genetic composition. This is called a genetic drift. Sixth factor for deterioration of variety is minor genetic variations. Minor genetic variations can occur even in varieties appearing phenotypically uniform and homogeneous when released. It is not of much importance, however, some minor genetic changes may occur during production cycles due to difference in environment. Due to these changes, the yields may be affected, and to avoid such minor genetic variations, periodic testing of varieties must be done from breeder seed and nucleus seed. The yield trials of lines propagated from plants of breeder seed to maintain the purity of self-pollinated crop varieties can overcome these minor variations. In self-pollinated crops, minor genetic variation is common feature. In often cross-pollinated species, due care during the maintenance of nucleus and breeder seed of cross-fertilized, cross-pollinated varieties of crop is necessary. Therefore, care should be taken during maintenance of nucleus and breeder seed. Seventh factor for deterioration is selective influence of pest and diseases. New crop varieties often are susceptible to newer races of pests and diseases caused by obligate parasites, and the selectively influence deterioration. The vegetatively propagated stock also can deteriorate quickly if infected by virus, fungi or bacteria. Seed production under strict disease-free conditions is therefore essential. Proper plant protection measures must be taken against major pests and diseases, otherwise the plant as well as the seeds get infected. In case of foliar diseases, the size of the seed gets affected due to poor supply of carbohydrates from infected photosynthetic tissues. In case of seeds and soil-borne diseases like downy mildew and ergot of jowar, smut of bajra and bunt of wheat, it is dangerous to use seeds for commercial purpose once the crop gets infected. New crop varieties may often become susceptible to new races of diseases or out of seed production programs. Another factor responsible for deterioration of varietal genetic purity is techniques of the breeder. Serious instabilities may occur in varieties owing to cytogenetic irregularities in the form of improper assessment in the release of new varieties. Premature release of varieties, still segregating for resistance and susceptibility to diseases or other factors can cause significant deterioration of varieties. When Sonalika and Kalyan Sona wheat varieties were released in India for commercial cultivation, the genetic variability in both the varieties was still in flowering stage, and several secondary selections were made by the breeders. This failure can be attributed to the variety testing program. In addition to these factors, other heritable variations due to recombinations and polyploidinization may also take place in varieties during seed production, which can be avoided by periodical selection during maintenance of the seed stock. Next factor is breakdown of male sterility. Generally in hybrid seed production, if there is any breakdown of male sterility, it may lead to a mixture of F1 hybrids and selfers. Another factor is improper seed certification. It is not a factor that deteriorates the crops variety, but is there if any lacuna in any of the above factors, and if it is not being checked, it may lead to deterioration of crop varieties. So this presentation was all about deterioration of varietal genetic purity. Hope this will be useful to all of you. Thank you very much. I have given here my YouTube channel details, Journey with Vijay Kumar Shrivastav. Having request, please visit the channel, subscribe it, and provide your kind and valuable feedbacks. Thank you.